My name is Ernesto Alfaro Moreno. I am the leader of the SiNfONiA project at INL. In the SiNfONiA project, we aim to develop the competencies related to nanosafety. Among the many different aspects of nanosafety, we aim to develop different aspects such as organ-on-a-chip, nanotoxicology, in silico assessment, and also epigenetics and bridging in vitro in vivo models using C. elegans as a model.
Under SiNfONiA project, my role is to establish the model to evaluate mechanism of toxicity and mechanism of adverse outcome of nanomaterials. Currently, within our two European projects, other two ongoing European projects, LEARN and iCare, we are partly engaging ourselves in the neuro-nanosafety. To address this neuro-nanosafety, we employed the nematode model called C. elegans. This is a microscopic model. It has no ethical concerns, and we can employ it for our genetic assays and neurological assays because all the neuronal mapping is done in these organisms, and almost 65% of gene is conserved from human to these worms. We address three physiological endpoints that is very critical to assess, like development, growth, and locomotion behavior. We have already established all these assays in this model, and now our aspiration goes beyond that, and we want to link it with this traditional behavioral assays with the neurological disorders and neurodegenerations. First, for neurodegenerations, we targeted dopaminergic neurodegenerations and glutamatergic neurodegenerations because that's the reason of choosing these, or selection of these are very compelling, because dopaminergic neurodegeneration is directly linked to the Parkinson's disease, and while glutamatergic neurodegenerations is associated with Alzheimer's disease. To assess these neurodegenerations, we employed a specific type of transgenic C. elegans strains for each different type. And in those strains, transgenic strains, we can actually observe and visualize their neuronal distributions in their whole body, and we are able to identify any kind of damage or deformations within them. To make it the next step, we adopted a scoring method for quantitative analysis, and that quantitative analysis is actually based on that phenotypic observations what we made in that qualitative assessment on those transgenic strains. Moving forward, we are aiming for future that we are going to establish Alzheimer's disease specific strains and with a specific type of behavioral attached to them, and then we want to compare with that with the cognitive one, and then with the wild type one. The challenge is here, there is no specific cognitive behavioral assays is developed or specifically for this model. By adopting all these approaches, we can ensuring neuro-nanosafety to actively achieve neuro-nano biocompatibility.